proteins in there that hold that chromosome together, so we have to give it a different name. Super loosely used terms, chromosome and DNA, so not a big deal, but you should know there is a difference. A gene, we know, is a small part of a chromosome. We know genes code for proteins, and proteins give us traits, and our traits determine basically everything about us. The first thing we're going to study is Mendelian genetics. All that Mendel did was discover the difference between dominant and recessive inheritance. You do need to know a few things about Gregor Mendel. Specifically, he was a monk, and you should know that back in the time when he was living, the only way you could get a good education was to go through the church. All the universities were owned and run by the Catholic Church, and so he went through that system so that he could get educated. And while he's hanging out doing his monk thing, he is working in the garden and started noticing inheritance patterns in the pea plants. So you need to know his name, that he was a monk. We call him the father of modern genetics, and he discovered everything by watching traits in pea plants. Before Mendel came along, people thought that all of our traits would be blended, which means by now every single human should look exactly the same. We know that isn't true, and because of Mendel, we have proof of how inheritance works. Mendel basically came up with two laws. If you think about the name, it helps you learn what the law means. To segregate means to separate. So the law of segregation tells us that traits separate into sex cells. And what that means is if you look at your DNA, you have two chromosomes for every trait. You got one from your mom and one from your dad. So you have 23 pairs of chromosomes. This represents one pair. So let's pretend from your dad you inherited brown hair, and from your mom you inherited blonde hair. You end up showing that you have brown hair because we'll learn about brown being dominant over blonde. But when your body makes a sex cell, you'll take one of those chromosomes and it will go into one sex cell. The other chromosome with that trait goes into the other sex cell. And then those become the traits that you pass on to your babies. So the two forms of a trait separate into different sex cells. I have those S's highlighted. That's a good way to remember that law. Here's another way to look at it. It's super impressive because Mendel didn't know about chromosomes, but he did realize that there's something separating into these sex cells that carries the trait. The other law of Mendel is called independent assortment. Again, think about what that word means and it helps you know what it implies. And that just tells us that there are lots of traits that make up an organism. For example, just because your dad has brown hair and brown eyes and he's tall, it doesn't mean that you will also get all three of those traits. You might be tall and have brown hair, but not get his brown eyes. So that tells us that just because genes come from one parent, they're not all stuck together. So you don't look exactly like your dad or exactly like your mom. Those genes are independent and they mix up differently so that babies can get different combinations of mom and dad. I'm going to skip this slide because it's weird. Vocab is super important. Make sure you know that dominant means that it will show up. It is noted with a capital letter. Recessive traits are wimpier, so they are noted with a lowercase letter. An allele is the fancy word for what is that trait. So for example, you can have a dominant trait for tall and a recessive trait for tall, which would be short. And then we put those into a Punnett square. So a Punnett square actually uses Mendel's laws. This would be the two forms of a trait that dad has. One form goes into a sex cell. The other form of a trait goes into a sex cell. These are the traits from mom. One form goes into this sex cell. The other form of the trait goes into this sex cell. When mom and dad get together, these are the types of babies that could come out of this cross. So Punnett squares show genes from one parent, genes from another parent, and the inside of the Punnett square is the offspring. More vocab. The letters that we use tell us what are in the gene, so that's called a genotype. When you talk about what that organism looks like, that's called the phenotype. So I will ask you to find the genotype of an organism, and then I will ask you to tell me what does that organism look like. So if you have genes that are both dominant, you're obviously going to show that dominant trait. If you get one of each, you're still going to show that dominant trait. If you have both recessives, you'll show the recessive trait. So if you have a genotype of big T, your phenotype is tall. If you have a genotype of big little, your phenotype is still tall because T is dominant. If you have a genotype of little t's, your phenotype is short. So phenotype tells us your physical, so pH and pH, or what you look like walking down the street. Genotype is what's actually in your cell. An F1 generation is the first generation, so you can remember first as in the first generation of a cross. That F actually stands for filial. You are your parents' F1s. When you have kids, your kids are your parents' F2s. If you want to be super smart when you go home today, you can tell your brother or sister that they're a stupid F1, and your parents are going to think you're swearing at them, but you're simply calling them your parents' offspring. You are also an F1, but I won't call you stupid. You're an awesome F1. So then we need to learn how to make Punnett squares. Again, you put the two traits that come from the dad. These are different sex cells. These would be the traits from mom. And then you simply take that trait and cross it down. If it's on the top, if you write it on the side, you write it in the squares going across. Now you end up with every organism needs two forms of a trait. So this could be a baby, here's a baby, here's a baby, and here's a baby. So when dad and mom get together, these are the four options of babies they could make. And then we talk about genotype, ratio, and phenotype. The ratio of genes, look at how many squares have big T, big T. In this example, one of the four, or 25%. How many squares have big little? Well, two of the four do. So two of the four, or 25%, have this genotype, and one of the four have little t, little t. When you are given or asked for a phenotype ratio, you tell me what they look like. So when this kid's walking down the street, they're tall. When this kid's walking down the street, they're tall. This kid is also tall. This kid is short. So if we don't know what's inside their DNA, we do know what they look like. So three of those four offspring are tall. One of the four are short. So genotype ratios, you tell me the letters. Phenotype, you tell me what the trait looks like. Some more vocab for you. Remember the prefix homo means the same. So if you are a homozygote, which means sex cell, a homozygote dominant, you have both dominant genes. If you are a homozygous recessive, you have the same and they are the recessive trait. If you are homozygous, you are said to be a purebred. Hetero means different. The only heterozygous we have is big and little. I can call this heterozygous dominant because dominant will show up in the phenotype. This was a typo. Your note should say hetero here. You cannot say heterozygous recessive. It doesn't make any sense because if you have a heterozygous genotype, dominant shows up. So you can only describe it as a dominant heterozygous. So don't use heterozygous recessive in the same phrase there. If you are heterozygous, you are said to be a hybrid, and we'll talk about that stuff later. So just plant that seed in your head for vocab. All right, I want you guys to try it. In your notes, see if you can cross a homozygous dominant with a homozygous recessive. What letters will be up here to be homozygous dominant? What go down here to be homozygous recessive? And then try two heterozygous parents. Tell me what the genotype and phenotype ratios are. So let's try these. Pause if you need to. And it should be this. Homozygous dominant, they cross down. The recessive traits you'll write over. Every single baby looks the same. So the genotype on this first cross is 100% big T, little t, and every single kid walking down the street looks tall. When I cross heterozygous parents, again, take that big T, bring it down. These little t's come down. This big T goes across. This little t goes across. It doesn't matter what you put first. I could write little t, big T here. I usually put the big T first because it looks nicer, but the order of these T's doesn't matter. It's just the fact that you have one of each. Then when I write the genotype ratio, how many squares are big T? There's one, or 25%. Go to the next square. How many squares match this one? Two of the four, or 25%. I'm sorry, 50%, two of the four. And then how many squares are little t? One of the four, or 25%. When these kids are walking down the street, these three will look tall. So phenotype is 75% tall, genotype is 25% short. And we can get a little bit crazier. We can do a two-factor cross because there's obviously more than one gene that makes up an organism. So let's pretend your dad has brown hair and brown eyes. Maybe your mom has blonde hair and blue eyes. But when they make their traits, you have to look at their genes. One sex cell could have big A, big B, another one little a, big, one big A, little b, and then the little little. So you look at the genes in the parents, and you have to make all the possibilities. So sex cells, then again, you just drop them down, bring them across, and you end up with this type of Punnett square. You don't have to be able to do this. You do need to do the single factor cross, but if it's easy for you, you should try this. Everybody has to try it at least. And then again, you've got to find genotype and phenotype ratios. So look at the first genotype, see how many squares look exactly like this. There's actually only one. So a genotype ratio would say one of these squares is big A, big A, big B, big B. And then you go down and you find how many match this, how many match this. So 2 of the 16 have this genotype, or 1/8. 2 of the 16 like that. 4 of the 16 look like that, and so on and so on. Then you look at phenotypes. If you have a big A, you have brown hair. If you have a big B, you have brown eyes. So how many squares have one big A and one big B? 9 of the 16 do. How many have one big B and one big A and two little Bs? How many have two little A's and one big B? And how many have everything little? So then you find what those kids look like. So genotype ratios, you count similar genes, phenotype ratios, similar what they look like. This is how Mendel actually figured it out with peas. So he did all of this independent assortment and segregation into sex cells. And if you think about it, there are hundreds of genes that make up a human. If you tried to make a Punnett square of what your actual sex cells look like, they would look like this, but there would be about 1,000 letters up here, which means it could go about 1,000 squares across and 1,000 squares down, and it would be a big, huge, crazy thing to make. So we only look at one or two traits at a time.